So, today's first session, biological crop nutrition, current status and the way forward. Each session, we have 5 sessions as you have seen, would be moderated by one of the BIPA's executive committee members who are experts in their fields. Now, for the first session, I invite Dr. K.R. Reddy, President BIPA, to please take on the stage and invite Dr. Reddy's respective speakers and probably introduce them also.
Good morning. So as our Executive Director briefed you, so we have 5 technical sessions today, and this is the first session.
Sorry to interrupt. Pointer to change the slides.
Okay.
Front, back.
Yeah.
Later.
Yeah. So we will be starting the first session, and I will be requesting the subject specialist and of course masters in their own academic activities to start with Professor Apparao Garu. Professor Apparao Padali is the Senior Professor of Plant Sciences, School of Life Sciences, University of Hyderabad, and he served as the Vice Chancellor of University of Hyderabad. Please come on to the dais, please. Next, Dr. A.K. Saxena. He is a Director and the Director of National Bureau of Agriculturally Important Microbes. Am I correct, sir? Yeah. Please welcome, sir. And we have Mr. Roger Tripathi, Managing Director of Global BioAgri Innovations, USA. Roger, please. And we have Aqua Agri Director, Mr. Tanmay Seth. Tanmay, please come on to the dais. And including me, we will be 5 speakers for the day. And the theme is, as it is mentioned, the BioAgri theme for this year is Bio-Innovations for Regenerative Agriculture. So why this regenerative agriculture is a hot topic today, we all know because we heard from stalwarts yesterday, including Dr. Ratan Lal. So regeneration is the very important aspect to be noticed by whole agricultural community, not only community, but also the environmentalists across the globe. So to reduce the temperatures, to reduce the carbon dioxide or carbon monoxide from the air, and also to regenerate the soils. So it's a dual advantage of regeneration. One is to produce sufficient food for the growing population, and apart from that, the quality food. So not only the producing abundant, but also it should be the qualitatively fit for the human welfare. So with this thought, we have actually 5 schemes, 5 sessions here. The first session is about the soil health and also the crop nutrition. So we all know here 90% of the audience are from companies. They're all keen to invest and also produce biological agri inputs. To start with, we have, as in today, the bio-stimulants, bio-fertilizers, and bio-pesticides, 3 categories. And recently, introduced the Act bio-stimulants generated curiosity and also, of course, past tremors across the industrial community. Getting the certifications are also G2, whatever maybe G3. These are all the issues will be discussed at the last session under the regulatory and also the business sessions. But here in this case, biostimulants or biofertilizers or biopesticides, but here in this case, we are talking about 2 aspects. One is organic biological inputs and microbial biological inputs. These 2 are 2 different categories. Organics, we know pretty well, they are all biologically derived. They are not living. The second component is the living component. They're all the microbial biological fertilizer, maybe microbial biostimulant or the microbial biofertilizers. So likewise, we have the organic biostimulants. They are derived from either seaweed extracts, amino acids, or humic acids, fulvic acids, or derived from any microorganisms. So these are the two categories, and we have actually 5 speakers will be dealing with all the kinds of biological stimulants or biological fertilizers, which helps in regeneration of the soil and also the plant health and plant growth. So with this, I think we'll be starting the first session with first speaker of the day, Dr. Aparag Aru. So he'll be talking about the plant microbial interactions and how they help the crop productivity and what are the business insights. Please, sir.
Thank you, Professor Dr. KRK. Good morning to everyone here. It is a very, very special meeting for people like me who are always in academics. And whatever we do, little related to industry, it is only after we have interacted with people like Dr. KRK and other friends around. I just wanted to share maybe a couple of experiences and also share some information which will be helpful for us to design and make use of kind of new generation microbial inputs for sustainable agriculture. I'd like to request the slides to be shown. The 2 experiences that I would like to share, because they are relevant, I wanted to share in the beginning itself. As early as 1984, when I was a PhD student, when we were looking for the wilt-causing bacteria, or trying to isolate the wilt-causing bacteria from tomato and other solanaceous crops. Every time, not every time at least, many times we ended up in isolating organisms like Enterobacter cloacae. And we used to send, those days we were not having NBIM, we were not having other facilities like IMTech, and several other things were not there. We used to send our cultures to a famous plant bacteriologist by name Bradbury. And every time we were surprised to see that the organism was identified as Enterobacter cloacae, which has hardly anything to do with the sample that we have collected. We used to think like that. But today we know the soil, the insects around, and everything that is around the plant, and including the human beings and animals around, constitute the agro-ecosystem. You cannot see them in isolation. That is one important thing that we have learned. Maybe we have not learned this at that time. It would have been a wonderful thing if we have paid attention to why there was Enterobacter cloacae all the time. Taking that as an example, and I would like to also tell something more, that we have started our journey working with individual strains, and we thought we were doing a good job, and things were accepted, and it was very well recognized everywhere. But as the complexity of the systems we try to understand, we realize now, working in isolation, working with individual cultures has less of a relevance in an agro-ecosystem, and we have to have a holistic approach where we are trying to talk today the plant as a holobiont, where you talk about several other things that are associated, and each one of us are acknowledging the importance of all other biological and abiotic factors associated with the plant to be responsible in modulating the growth, in modulating the yield, and also in fighting against the pathogens. With these 2 examples, I would like to take you through a few slides that I have put together to share my thoughts on this occasion, where some of you may feel that it may be, part of the information may be useful for you. I said next generation microbial inputs for sustainable agriculture. That is about 3.5 years ago, when I was in MIT, I was interacting with several startups. And several of them, one after the other, they were all working on human microbiome. And I'm sure there are several products which many of you know by now in the market about centered around the human microbiome related studies. But the work on plant microbiome has started little later than human microbiome, and we are now in a stage where we can think of newer products. I will just take you through the story of what it is. Yeah, he reminds me about the time. Professors will normally take a little longer, Dr. KRK. These are the different types of organisms around our plants, and we cannot see only plant in isolation or a single organism in isolation. This is what I wanted to convey with this slide. And this has not changed, KRK. I think they have not put the latest one. Okay. Can anybody help me to go back to the latest one? No, otherwise, don't worry about it. We will go through this. This is I have actually added a few slides this morning after I interacted. What I wanted to tell there is in this agro-ecosystem, our agricultural practices can significantly contribute, as many of you know, can significantly contribute to alter the microbial community structure and function. This is what has been realized in many different ways with several different crops. And when you realize that there is a possibility of change, then how do you regenerate? This is what I think Professor Dr. KRK is interested. Regenerative agriculture is what he's interested in, and he's going to talk about it. And how do we regenerate is the question. And to regenerate, we need to understand the details of the community structure and how this function of this community is altering with different agricultural practices. And we know some of them by now, and some of them are yet to be known. We know the diversity is changing, the composition is changing, the numbers are changing, but we do not know functionally how much this has relevance. Some of them may be relevant, some of them may not be relevant. That is something which we need to understand. Another important thing that we need to understand is that these microbial communities, in the name of synthetic communities, are coming up. And can we really push them sufficiently to make a desired alteration in the community structure? That would be the next generation of next generation of products that will be coming beyond the synthetic communities. I will be taking example towards the end about the synthetic biology and as to how that is contributing here. Last important point that I would like to mention before I go through the slides is, yesterday in one of the presentations it was shown, very recent information shows that the BioAgri inputs, the three Bs that all of you talk about, market is around $6 billion US dollars. There is an estimate now, I'm sure many of you know about it, by 2023, just in 2 years from now, that market is likely to grow to $14 billion, just about to double in about 2 years' time. That means the buzz and the focus on developing these kind of products for sustainable agriculture has been at a rapid pace. And that is what is going to be attractive for all of us sitting in this room, and for us as academicians and for you as industry people. They are the ones which will drive us to develop the new products. When you ask for this, there is do we have such kind of products in the market? Do we have such kind of established products which are tested? And where you know the details as to how they are working? Unfortunately, we are yet to have some clear understanding about several of them, excepting a few cases where we have defined information about those communities that we can synthesize and then introduce them into an ecosystem, an agro-ecosystem, and then take advantage of it. So
please.
Opportunities are plenty.
You have another 3 or 4 minutes, sir, please.
Opportunities are plenty. We can go ahead and then look forward for such kind of things. Those are the different ways in which the epiphytic and endophytic microorganisms interactions take place. These are not new to all of you. All of us sitting in this room are quite familiar with what are all there in these slides. That's where I was not emphasizing on that. There are several different ways in these nutrient transformations or acquisition pathways are linked to plant and microbe interactions. And several of them can be manipulated. And you have these, on one hand, you have these components like insect, plant, and soil. On the other hand, it is also connected to the crop-associated microbiomes. They are shaped by the agricultural practices. This is the point which I was mentioning in the beginning. I was also telling you that so far we have been looking at the left-hand side, the reductionist approach. Although we made some progress, in terms of moving from single isolate to stacking some of the consortia, and now it is the synthetic communities and the microbiome towards a holistic approach that we would be looking forward. And it is this kind of a dynamics that one needs to understand. Understanding the individual metabolomics, proteomics, genomics is much easier, and understanding it at community level is more complicated. And if we can understand, and that's where the modern biology like systems biology and synthetic biology are coming into picture, where we can draw metabolic networks as to how each one of them are going to influence. That is going to help us to understand how these next generation microbiome products can help us to do the wonderful jobs. And these are certain things that how these are the different plant-associated microbiome functions which can help the plant in better growth and better nutrition, both by direct and indirect mechanisms. These are the last few slides, like future of microbiome-informed management. This is given in terms of, it is not exactly a time scale, first generation, second generation, third generation, but what we see is all these dotted lines are yet to be connected. Please remember, we are yet to connect these dotted lines, and where we only have this kind of an information so far available. And once we connect all these dotted lines, we will be moving towards the third generation and future precision microbiome management based products. That is going to be more useful for us for sustainable agriculture. And yesterday also it is talked, and all of us realize the importance of, to attain some of these approaches, like this in indoor farming or in the greenhouse, for these how much of a microbial inoculants, what is the role of the host, and the predictive analytics, the big data that comes into picture, which comes through these studies of community level interactions, all of them will help if there is a partnership between the academia, growers, and industry towards an integrative approach. And this is what I was trying to bring in. And I think last slide. These are the synthetic biology components that have emerged in the recent past. When we look at the plants, several creative concepts are being looked into. Some of them include the synthetic microbial communities. I'm not bothering you with several other things that are there about the plant. All of them are relevant for us. If somebody wants to look at the plant itself, all of them are relevant. And if we can bring them into the next level, that is to the proven possibilities. If we can bring them to the proven possibilities, we will be able to take them commercial applications. And these are the ways in which the startups are looking globally. The startups are looking globally, making use of the synthetic biology approaches. So with this, my dear friends, I don't want to spend more time and take the liberty of taking extra time. I want to only convey that a better academia, grower, and industry interactions with this kind of a holistic approach, and if we look at the knowledge-driven approach, knowledge-driven science, knowledge-driven product development, we will have a better sustainability. And as yesterday it was mentioned very clearly, a One Health concept is something what we have to keep in mind. Thank you very much. And these are some of my students who have been with me and in shaping some of our research in the lab. And I also need to thank the financial support that I've been receiving from the Government of India for different projects. And finally, thanks to all these BioAgri Input Product Producers Association and the organizations of BioAgri 2021. Thank you so much.
Thank you, sir. I know the constraint. The time is the essence of the day because we have almost 20 presentations on whole day covering in 5 sessions. Therefore, I request not only the experts on the dais, but also the future speakers kindly restrict to 10 minutes. And it's a very interesting, and this particular technology, microbiome, is the future of agriculture. So now I request Dr. A.K. Saxena to speak on the microbes for the betterment of soil health. Dr. Saxena.
A very good morning to all of you. First of all, I would like to thank the organizers for inviting me for this very important conference. As has been mentioned, I am working as a director of National Bureau of Agriculturally Important Microorganisms. It is a place where we conserve and preserve the agriculturally important microorganisms, particularly those which are used for bio-fertilizers, bio-pesticides, for bioremediation, for composting, and many different activities which the microbes can do for agriculture. I think many of the biopesticide producers who are sitting in this hall must be aware of that now for any cultures to be used for biopesticides, the DNA fingerprinting has to be carried out, and that is being done at NBIM Mau. Today I'll be talking to you about the microbes, their role in plant nutrition. And can I have my slides, please? Okay. So whenever we talk about the plant nutrition, you see the microbes have a lot of potential. They are the ones which are basically involved in many of the nutrient cycling. And when we say nutrient cycling, that means the plants, they take up different nutrients in different ion forms. So it is basically the microbes which are able to provide the nutrition in the form in which the plant can take up. When we talk of nitrogen, it is basically the nitrate or the ammonia which is being taken up by the plants. And when we talk about phosphorus, it is the orthophosphate or the phosphate form in which the plants can take phosphorus. Likewise, for potash also, the ionic form is being taken up. Zinc is taken up as zinc. Then sulfur, if the plant has to take, it is taken up in the form of sulfate. So these are the different ionic forms in which the microorganisms, they help, and they help the plants to take up different nutrition. Now, yesterday, when you were listening to Pan, she mentioned about that how to sell the product. And the first point which she mentioned was one should know the mechanisms by which these organisms actually work so that you can make the people aware of how these organisms are working. So here I have listed some of the mechanisms which are being pursued by the microorganisms for carrying out these activities through which they provide the nutrition. The first of it is the nitrogen fixation, and we all know that it is only the bacteria, the prokaryotes, which have the capacity to take the nitrogen from the atmosphere and convert it into ammonia and then give it to the plants. The second mechanism which the microorganism normally follows is the solubilization. When we talk about solubilization, it means that many of the nutrients like phosphorus, potash, and zinc, they become unavailable to the plants because they get fixed in the soil. That is why when we say the use efficiency of phosphorus is only 20%, that means if you are applying about 100 kg of phosphorus to the soil, 80 kg gets fixed in the soil, and only 20% is available to the plants. So these are the microorganisms which carry out the solubilization activity by producing the organic acids, and then they make the phosphorus available to the plants. The same procedure is for potash and zinc. Here again, the organic acids are being implicated in solubilization of these nutrients and then making it available to the plants. In addition to this, ACCA deaminase enzyme, which is also responsible for enhancement of the plant growth, not directly in the nutrition. Then we have other mechanisms like production of many of the plant hormones like indole acetic acid, gibberellic acid, cytokinins. These are the hormones which normally the plant produce themselves to enhance their growth. But there are microorganisms which have the ability to produce these plant hormones, and they also help the plant to grow in a better way. So these mechanisms actually are the ones which are carried out by most of the bacteria and the fungi on which we work and through which we develop different formulations. Can we move ahead, please? Yeah. So when we talk of these organisms, if you look at the table, you will find that Rhizobium, Azotobacter, Azoospermum, Gluconoacetobacter, Cyanobacteria, these are all nitrogen-fixing bacteria, and many of the producers who are sitting here, they must be using these organisms for making the formulations, whether it is a liquid-based formulation or a carrier-based formulation. And these organisms, they can provide about 25 to 30 kg of nitrogen per hectare to the plant. Among these nitrogen fixers, if you look at Azoospermum, which is the one which is the least used by many of the producers and which is not produced on a larger scale. But here I would like to mention that this is the only inoculant which has an optimum temperature of 37 degrees centigrade. And we should not forget that this is one of the tropical country, India, where the temperature sometimes goes as high as 40 degrees centigrade. So this becomes a very important inoculant to be used, particularly in areas like Rajasthan and Gujarat, where the temperatures are high. And this organism can work very well. Although it is not a very efficient nitrogen fixer, but it is a very good producer of hormones. So it can improve the growth of the plants, particularly the millet crops and some of the cereal crops. Then we have the P solubilizers, the K solubilizers, and the zinc solubilizers. Here in the FCO, if you look, this fertilizer control order gives the complete group. That is, here no organism is mentioned. It is only the PSBs, the KSBs, and the ZSB. Here one has to be very careful when you are producing the product as PSB, KSB, or ZSB, that it should not have any harmful effect to not only to the resident microflora, but also to the plant. Then the last one is the AM fungi and the piriforma. AM fungi, yesterday also we heard about. It is one of the very, very good inoculant. The only problem is that it is an obligate endophyte, cannot be cultured, but it has to be maintained on the plant roots, or it has to be grown, the spores are grown using the culture techniques. Can you move forward, please? Now, this is one of the consortia which was released in the Gazette of India in 2014. This is the only consortia available under FCO, and here it is the bio-NPK formulation. Now, this particular unique formulation basically provides all the three nutrients to the plant, whether it is phosphorus, potash, or nitrogen, and it can provide at least 1/3 of the nutritional requirement of the plant. So this is one of the very important formulation which has to be promoted on a larger scale. And here I would like to mention that this is one of the patented technology which has been developed by me. And here we are using only a single media for growing all the three cultures. Now, we know about bacteria, we know about fungi, but do we look beyond that? Do we look for other options also? So here I would like to give a brief view about the other organisms which can be looked for. Just move forward, please. Now, when we talk about the eubacteria, the fungi, we tend to forget about the third domain of life. Third domain of life, when we say it is the archaea. You see, the fungi belongs to the eukarya, that is one domain. The bacteria belongs to the prokarya, that is the second domain. And the third domain of life is archaea. Archaea has never been used as a plant growth promoting or for plant nutrition. So this is another domain which has to be looked for. So we have to look beyond the bacteria and the fungi. Can we have other options? Next, please. Now here I'm just giving you a brief about the run of Kutch, which is a place where a lot of salinity is there. Now when I visited this particular place, I found a lot of vegetation in this area. And the question immediately came to mind, how this vegetation is growing, what is sustaining this vegetation. So then I tried to look for whether there are archaea, because archaea are known to be primitive organisms, and they are present in the extreme environment, whether it is a low temperature, high salinity. So immediately I thought of whether the archaea can be an option for these plants to grow in these areas. Next, please. So from these areas, we isolated some of the archaea, and you can see the colorful colonies of these archaea which are growing in these extreme environments. And then we tried to look for whether these archaea can have any stimulation on the germination or not. That is the first step whenever we do science, to see whether the organism has any beneficial effect or not. So we found that many of the archaea, they are inhibitory to the germination of the seeds, but only some of the archaea, they were stimulatory. So we carried out our work further on this, and we found that these archaea, just move forward, please. These archaea, they also have similar functions just like the bacteria. They are good producers of IA. They can also solubilize P. They can solubilize K. They can produce siderophores. They can produce ammonia and HCN. So some of the attributes are basically for biocontrol, like the production of HCN and ammonia. But some of the attributes are basically for the plant nutrition. And if you look at the range of the P solubilization, the eubacteria, which we normally use for the solubilization P, the range is only 11 to 520 milligram per liter. FCO only wants that the culture should have 30 mg per liter solubilization capacity. But if you look at the archaea, the range is much higher, 1986. That is 4 times more than the eubacteria. So we should try to look for these options also, whether we can bring it into formulations and whether we can use it as a biofertilizer. Next, please. So these organisms, they have been tested, and we found that some of these bacteria, they not only helps these archaea, they helps the plant to grow better, but they change the anatomy of the plant. They also act at the gene level to initiate some of the activities. Next. So we are also looking for these archaea for alleviation of abiotic stresses, particularly the drought stress and the salinity stress. And they have been found to be very good because we can grow the wheat crop from seed to seed without giving any irrigation, although the yields are much lower. So these are some of the options which are available. And here, finally, I would like to say that the biologicals have answers to many questions. It is now the need of the hour to look beyond. Thank you very much.
Thank you, Dr. Saxena. I know the subject is vast and the opportunities are huge. So now I request Mr. Rosa Tripathi from BioAgri Innovations to speak on the biostimulants and the current status, maybe the world status. Rosa.
Thank you, KRK. First of all, Dr. Saxena, I like your passion and persistence KRK was standing up, he was ringing the bell and everything, but Dr. Saxena was so persistent, he kept going with his slides, so good message. Second thing, I really want to congratulate BIPA for this organization. The first time I heard about this was 6 weeks ago, and the job you have done is fantastic. As you know, I have spoken at different events. The energy I felt yesterday, the knowledge here, I really want to congratulate KRK and the committee and Raj. Thank you so much. One name was mentioned a lot yesterday and today, Akash. Whoever Akash is, seems to be a magic man, everything Akash. So thank you, Akash, for that. One thing I also liked about, can you put my presentation, Akash? Next. Where's the pointer? Okay, so it will work for me? Okay. So this is a little bit about myself. A lot of people were asking yesterday about my background. So I'm OCI or Indian at birth. A lot of people in this room, yesterday was like a reunion for me. So we have worked in the past and we have been together. And then from here I moved to Australia, became Australian. And then I moved to the US. This is perfect time for Dr. V's family to be here. And then the most important thing is that I have worked in almost 45 countries. So today what I'm going to share a little bit beyond the scope of this presentation. I have been hearing since yesterday what our industry in India has done. So I want to bring a little global perspective because I started as a scientist, became an agronomist, and then from there I became very hardcore commercial person. And that's when I started my own business 3 years ago. So this is the first of my business. The reason I'm sharing this, because a lot of people ask me, what do I do now? So this is the one of my business, Global BioAg Linkages. As the name says, that's exactly what we do. We connect BioAg businesses globally. And listening to Dr. Saxena, Dr. Rao yesterday, if you look at what we have in India, we can be the next hub, especially in the bioag industry. In any case, we are a global community. What we have to look at how we expend this knowledge globally, not only for commercial value, but also for sustainable agriculture. In this regard, as you have seen Pam Marone yesterday, she did the presentation. She's also a partner in the business. Then we have different other verticals. One of the vertical is go-to-market, where we help a lot of businesses to go to market globally. We have also one event, which I will talk a little bit later, BioAg World Congress, that we organize. Eva is managing that. When global businesses are expanding, they are looking for a specialist recruitment. So they come to us and we organize a specialist recruitment through Nancy. She is the guru. She has done that for 25 years in the bioag industry. Then regulatory, which is managed by Rejane, a Brazilian-American, PhD from DuPont. She manages that vertical. And then we also started a magazine, BioAg World Digest. This is the second business. The first one is service-oriented and more consulting, networking, connecting, linkages. The second one, being in the product business all my life, I could not keep myself away from the product commercialization. And if I ask you how many businesses in US in the bioag are making profits, you will be astonished. Not many, even though we have a lot of businesses globally. Therefore, I decided to create a very unique commercialization platform where we bring the innovation from two pipelines. One, the young, dynamic innovators such as you, who come to us and they say, we don't want to commercialize globally, it's too much of a hassle, we will use your platform. And the second one, where Pam and I get together, we develop our own recipe and formulation with different technology platforms, and then we get that formulated with our established innovators across the globe. This is how we get our innovation pipeline. The entire model is based on partnership. Coming back to the biological products, I don't know if you remember this slide 3 years ago was very different. 3 years ago, biological control was shown as a bigger piece. But today, the biostimulant or the crop improvement area has caught up, and we have the same biological control and the crop improvement area almost 50-50. Yesterday, we were also talking about the biochemicals and the microbials. In a lot of industries, what people are doing these days, they are not separating it. They are making synergistic products from the microorganisms, macroorganisms, and biochemicals with microbials. With regards to the bio-fertilizers and biostimulants, the most important thing what we have to understand, as we heard yesterday, how do we make it prevention is better than cure? And that's where I see our industry can again take the lead. If you look at all the things Dr. Saxena was talking about, how we make the nutrient more available to the plant, that's where we have to go. We have already used too many chemicals, too many products, too many inputs without thinking how much of that is taken by the plant. Therefore, what we have to do through the bioag industry is trying to make the availability of those nutrients to the plant, and the rest will take care of itself. If you look at how the plant health and the crop yield is managed, microbials has a big role to play in addition to the extracts, the acids, and other synergistic products. Most important thing is about the single microbes versus the consortia. I am a big believer the characterization of the single microbe is important, but if we can work on the consortia, as you were saying this morning, that is where the market is going. Because with the consortia, we can make our research more commercially viable, and we can solve more problems with one product than launching several products. The most important thing what we have to understand with the microbial biostimulants, which I think Hyderabad and India is becoming the hub, to understand these aspects, that this is the market which is growing extensively in all the regions. The entry of the new products and technologies is taking place, but there is a big challenge. One of these challenges, which I mentioned in Basel last week, was that they are stopping the chemicals. They are saying we need to bring the bioproducts, but at the same time, there is no regulatory framework. And if there is a regulatory framework, it's more creating barriers than trying to facilitate it. So what we have to find is a way to move into the globalization, harmonization of our regulatory platforms. All the regions in the microbial biostimulants are really at par. If you look at the North America, US and Canada, Europe, Asia Pacific, LATAM, they are all almost 40 to 50 million. In my opinion, this has changed dramatically. This was in 2019. I think this is rapidly growing, and we can take part in this in the global perspective. At the same time, you see here the US biostimulant market has 15% microbial, 85% non-microbial. Dr. V, I think you will agree that now this is more than 30%. It's almost doubled. And again, this is a great opportunity for us as an Indian industry. These are different microbial industry leaders, as you can see here. Agrinos, which is now sold to MVAC, BSF, Bayer, Bioconsortia, all of them are working in consortia than single characterization. These are the big players in the biostimulant industry globally, and there are hundreds and hundreds of more of them. What I liked since yesterday, we talked a lot about the soil and the foliar. And I personally feel that to manage the abiotic and biotic, we have to again look at both, the soil applied and the foliar applied. If you look at what are the things we are looking for is the site of interaction, plant physiology, modulation, nutrient uptake, which I talked about earlier, gene expression modulation at plant level, modulation of microbial community in the soil, and at the same time, the activation of microbial intervention. Last but not the least, this is very important. The reason I said last but not the least, so KRK does not nudge me. This is the last slide, KRK. Trust factor is the biggest factor. A lot of us know this snake oil image. And we have faced that in India and globally. This trust factor about the selfish profiteering in the short term has to stop if we want to bring any credibility to our business, our industry, which BIPA can play a big role in that. Last but not the least, now for sure, KRK, is I want to invite all of you to the BioAg World Congress in Valencia from 26th to 29th. And I put this slide only so that I can answer everyone's request from last night.
So how much discount you will give for BIPA members?
BIPA members, you will get 5% immediately. Okay, we will consider 10%. Here we go. Thank you so much, everyone.
Thank you. So there are two things. One is we may have to sacrifice time of panel discussion. And the second thing, there is nothing like a specific coffee break. Those who want, they can collect from outside. So now I welcome Tanmay Seth. He will be speaking on seaweeds, utilization, and his experience. Tanmay, please.
Thank you, sir. Very good morning to all the distinguished delegates. Thank you, BIPA, for giving me the opportunity to speak at this conference. We are a company very specific on cultivation and processing of seaweeds. So that's what I'm going to take you through. I think the knowledge bank available here is far more vast, but we are a focused company on seaweed processing, and I'll be sharing my experience with that. So seaweed, as most of you know, is a unique plant that grows in the ocean. It requires neither land, nor water, nor fertilizer. And it's proven to be a very viable biostimulant, which was very well quoted by Dr. Kalam. And there is a lot of emphasis now from the PMO and the Prime Minister's office on increasing seaweed cultivation in India, as it offers an opportunity to give employment to more than at least 100,000 people. I wanted to share the latest development of the sector. Very recently, under the Pradhan Mantri Matsya Sampada Yojana, 640 crores have been allocated for the development of this sector. We are one of the companies chosen to also lay down the roadmap, and the government aims to achieve production of 11.2 lakh metric ton by 2024-25. We are not even on the map today, so the opportunity is enormous. There's a lot of experts from different fields here, and I think one point I'd like to bring up is the seaweed cultivation in India has been based only on one species called Kappa ficus, and that seed has lost its vigor. And experts here can contribute in a very big way to bring that, why is the production gone down? How can we treat it with natural products to break the productivity back up? I think it's a very interesting area of research, which together we can put our minds on and solve. Seaweed aquaculture forms a very good strategy for India, for livelihood creation, for climate change. I think one major issue hitting us all these days is the amount of CO2 which is increasing and the methane emissions. And I think seaweed offers an opportunity to cannibalize and take up carbon dioxide, which is a very big opportunity. It helps in reducing ocean acidification, ocean deoxygenation, and it has very low investment requirements and offers an opportunity of livelihood for the coastal communities. I'll very quickly run through our journey. This project, the idea of seaweed cultivation started in '98 under PepsiCo. In 2008, BioAgri, our company, was formed. 2016-17, we had a tie-up with a fertilizer major, which is also an important point to highlight that the mindset is changing. The largest chemical producer and distributor in the country, Indian Farmers Fertilizer Cooperative, also looked at us, considered us, and now is a 50% partner in the business. And their mindset is changing from understanding the need to switch over to biologicals and natural products. And that's a very, very encouraging sign for all of us and the country. The algae we specifically work on is Carpa ficus alvarezi. It is cultivated on the Tamil Nadu coast. We have been trying to do it in other parts of India, but so far success has come only in Tamil Nadu, primarily because we operate in an area which is protected by Sri Lankan landmass. This is a quick visual overview of how seaweed is grown. It's a very simple process of taking a bamboo raft, which is 3 meters by 3 meters. We plant 60 kgs. It's a vegetatively propagated species. It's a 45-day cycle. At the end of the 45 days, farmers are able to harvest 300 kgs. They replant 60 kgs. Dry or wet biomass of 240 kgs is sold to us and other companies in this field. The beneficial impacts, a few have been highlighted. Dr. Reddy had asked me to highlight the process innovative technologies globally. So I think what has been unique to us, the technology license to us is from Central Salt and Marine Chemical Research Institute. Globally, all the work has been done on brown algae, kelp, and Ascophyllum nodosum. Most people believe that is the best species to produce biostimulants. I beg to differ. I think it's about investigating various species. There are equally good properties in tropical algae, and we have a very big opportunity to collect and grow those species in India. We can, looking at the costs of production, we can make biostimulants affordable to broad-acre crops, and the opportunity to do this completely indigenously exists, and it's for us to exploit. Biostimulants have proven to reduce chemical load in agriculture. We've done many studies to prove at least a 25% reduction in chemical fertilizer. There will be an opportunity to trade carbon credits, which I believe will offer a higher income source to our farmers who will get into cultivation. It creates a new habitat for marine organisms. We are doing some research on it. I have not seen it, but China has created systems with balanced cultivation of fish and seaweed where the oxygen-carbon balance is maintained to make the grower benefit from higher fish output, higher seaweed, in the end, making the activity more commercially viable. The red algae, one thing mentioning on the technology part, most brown algae process is based with alkali hydrolysis. So the potassium source is chemical-based. They have permitted KOH as a permissible processing aid in organic agriculture under the US NOP standards, not in India. But what is unique to red algae, which we have here, it in itself takes up about 30 to 35% dry weight of seaweed is potassium chloride. So our potassium source is 100% biological. It's derived from the ocean. The bioavailability of this is higher. So we believe a much smaller dose could do the job. I don't have time for showing you a clip, but I'm happy to share the link with anyone who's interested in what the activity looks like. Sorry. So it'll take up all the time. So the opportunities, biostimulant is only one such opportunity. There is hydrocolloid derivation from seaweeds. You can do agar, you can do carrageenan. There's various, you can do ulvan. There's a variety of hydrocolloids. India is an importer of these hydrocolloids. So seaweeds often are an opportunity of value addition in the country. There is a lot of interest right now on asparagopsis as an input for reducing methane emission in cattle. I believe India can offer an opportunity to grow it. We are also exploring the possibility of growing it in different parts of India. I think we could be the lowest cost producer and a global solution provider for that. There's nutraceutical, organic chemicals, biodegradable packaging, edible packaging. You may have seen the orb in the UK marathon where they developed that little ball on the last right-hand corner, which is an edible ball given to the marathon runners. This is again carrageenan based, which is derived from seaweed. The technology we have is from CSMCRI, which allows us to produce bio-stimulant and kappa carrageenan from the same biomass, making us far more competitive than the rest of the world. We have innovated on this technology and developed it far further from what was given to us. The brand, we're selling the product under the Sagarika brand name in the Indian market. We have seen very, very good acceptance of the product. It has high level of natural potassium, and it's a very integral part of balanced soil building program. It increases the beneficial microbes in the soil, allowing for better uptake of nutrition, impacting nutrient use efficiency very, very positively. Between 15 to 25% average increase in yield across India, across all states, all soil types, has been seen and proven. This is the range which we sell in India through IFCO, which is available in liquid, granular, water-soluble powder form. The product is certified organic as per Indian, US, Canadian standards. We are in process of building international relationships, but idea was to first exploit the opportunity within our country, reach our farmers, and then look elsewhere. Our growth focus going forward will also be to work on animal feed, fish feed, poultry feed, as immunity is becoming a subject of very high interest. There is a ban of antibiotics in this field, so I believe seaweed offers a very, very large opportunity to build immunity in animals, health in animals, and also impact methane emissions and carbon credits. Last, Dr. Reddy had asked me to include the regulatory status of biostimulants. I can share the experience of it's basically our experience for trying to get registration for seaweed-based biostimulants. Currently, there is a confusion on the issue of Form G2 in most states. I think as institutions like BIPA can help, we can, as an industry, formulate a standard suggestion of the parameters which should be put on the product, such as seaweed, humic acids, and others. We should first agree internally on a standard, present it to the government, so our chances of success will be higher. There is a challenge in defining the specification, so we need to put our minds to it. An alternative could be to propose truthful labeling and the test methods which are common to all. Last, we've got some awards on being recognized as a science and technology innovation for rural development. It has also been launched on Technology Day by the ex-President of India. And I'll conclude with that. Thank you.
Thank you, Tanmay. Excellent time management. Thank you so much. We are saving almost 2 to 3 minutes. I think this is my term. I will hand over bell to Rosa. So 8 plus 2 minutes. Be strict. Not now. Yeah. So may I have my presentation? So I will be speaking on microbiome engineering for regenerative agriculture. So engineering, it's not difficult to understand. Here the engineering is nothing but assemblage. So assemblage of various kinds of microorganisms to improve crop productivity. That is the basic idea of microbiome engineering. See, regenerative agriculture now, I think since yesterday evening, a lot of speakers are, I think they have intimated us how the soils are deteriorated, our natural resources are eroded. So here in this case, how to utilize the bio-innovations with respect to the microbials. So these are the various components of biological regeneration of our agriculture. So one is how to bring back life to the soil. So we lost really life out of the soil, and not only life, but also the various physical and also chemical essential parameters of the soil. So therefore, we need to restore again and also bring back all these things to the soil. So it's already mentioned by a lot of other speakers, like another 50, 60 years, we will be losing the productivity of the soils. Soils almost going to be die. So therefore, it is high time for us to implement various biological practices, organic practices to bring back life. That is the serious, in fact, actually, even globally, everyone is now talking about restoration of the properties of the soil. And at the same time, carbon sequestration. And we have a lot of carbon dioxide, and that is very essential for the plant growth and also productivity. Therefore, how to send back these airborne or maybe the carbon dioxide of the air into the oceans as well as into the soil. That is a task. So soil, yes, through agriculture we can definitely do. So these are the various things. One is the mitigation of abiotic and biotic factors. Now we are talking about microbiomes. What are they? I think we have two speakers. So learned speakers, Professor Aparagaru and Dr. Akhil Saxena, they talk about the various microbes. So these microbes are essential, these microbes are ubiquitous, and these microbes are present everywhere. And you can see the microbes associated with the human being, microbes associated with the insects, microbes associated with the plants. So therefore, these microbes associated with the plant, on the whole, they may survive inside the plant system, like in the vasculature. They may survive on the surface of the leaf, or maybe on the aerial parts of the plant, and also in the rhizosphere, free living are associated with the rhizosphere. All these microbes associated with the plant, wherever they are, collectively called as microbiome. So individually, all these years, most of the scientists are focusing on the individual aspects of nitrogen fixation, phosphate solubilization, likewise. Now it is a consortium-based applications where people are understanding the mechanisms, physiological, biochemical, and also molecular mechanisms of plant microbial interactions. Therefore, how best we can utilize consortia so to mitigate abiotic and biotic stresses apart from crop nutrition. So this is holistically the microbiome approach. And as I said, these are microbes associated with the various plant parts like phyllosphere, endosphere, rhizosphere. So likewise, and also insect microbiome also very, very, very important. We talk about entomopathogens. That means the microbials, especially the viral entomopathogens, bacterial, fungal, and also nematodes, they also kills the deadly insects which damages to the crop. So insect microbiome and also the plant microbiome, which are really the big business opportunities now. So you talk about any nutrition, as we mentioned about the 3 Bs again, the bio-stimulants, bio-pesticides, and also bio-fertilizers. All the three where we have the microbes are associated. Microbial biostimulants, and also we have another set of the experts here sitting here. They spoke about organic biostimulants. This side they spoke about microbial biostimulants, biofertilizers, biopesticides. So there are several direct and indirect actions of these things. Each action can be a business vertical. And so this is the basic platform, how to understand. So this is, once again, you require to establish the proof of concept and individual microbial functions, both as I mentioned about the physiological, biochemical, and molecular aspects, and then understanding and application of the omics. So they are, the phenomics, the genomics, the metabolomics, these are all the tools available. Therefore, now the research and development is easy. It's not the case 30, 40 years back. So now all these molecular tools, we can say that it's a kind of a soft toolbox is available to understand the function of each microorganism. Therefore, you can make a proper consortium into converting into a delivery system. And this is how the schematic representation of how to do a microbiome engineering. Understanding the core microbiomes or base microbiomes, that means what are the microbes which are essential to be there in the rhizosphere of every crop. Crop recruits its own microbes. Crop also packs their own microbes in the seeds. So likewise, the microbes also goes from generation to generation. So these microbes, if you understand what kind of microbes are associated with a specific crop, that means genome demanded microbial communities. This because some plants, they produce specific exudates through the root system into the soil. So those exudates attracts a set of microbes. That means there is an intimate connection between these exudates and the microbes. Therefore, we need to catch those microbes, and also what it's called, keystone genera or taxa. So likewise, we need to understand specific microbes associated with the crop and also general microbes with individual functions. These functions, we are talking about many functions. So these are the business verticals where you can exploit the microbes thoroughly for seed priming and soil amendments and also foliar sprays. Seeds priming, I think it's globally, business is growing, but still in India it is a nascent stage. But there is a great opportunity for developing seed priming delivery systems with the multi-microbial consortia. So these are the various opportunities, and botanical and marine extracts, enzymes, vitamins, amino acids, and humics all falls under organic biostimulants. And apart from that, we have the microbes, cell-free extracts, these are the metabolites, and these are also can be extracted from the microbial systems. And at the same time, we have the crop nutrition, disease management, and also insect pest management. So the 3 Bs, you can exploit these microbial communities. And of course, global status and also Indian status of regulations, we have FCO, 1985 and also Insect Act 1968. So where 1968 Insect Act covers bio-pesticides, all these viral, bacterial and fungal bio-pesticides. And at the same time, so we have the microbes under biostimulants as well as bio-fertilizers covered under FCO. Now the biostimulants, all of you know that what the industry is facing. Now I think we have clarity from the states that they are going to accept the applications. And so these are the key steps towards successful use of microbiomes and the tools for food and nutrient security. So these are the researchable areas. Each researchable area can be converted into a business vertical, as I already mentioned, and also these are the deliverables, and these presentations are available for all the audience here. So finally, this is what I want to speak here, finally. So this is the holistic schematic representation of exploiting the microbiomes in agriculture. You see here, so I have mentioned about, this is actually drawn by me, and you have 3 aspects. One is biotic stress, abiotic stress, and also the nutrition. So abiotic stress, so as well, Dr. Saxena also showed the last slide, where soil, sodicity, alkalinity, salinity. So this is increasing every year after the year. So because of excessive use of chemical fertilizers, not only fertilizers but also several chemical pesticides also including the weedicides. All these things are all deposited into the soil, and the soil physiochemical structure is changing from year after the year. Therefore, we need to bring once again the changes in the soil to support the plant productivity. So therefore, the abiotics apart from the salt stress, we have like drought stress that is also increasing. Therefore, these microorganisms plays greater role in alleviating and also mitigating both salt and abiotic stress. And biotic stress like disease management and also insect pest management. Here, coming here. Disease management and insect management, where effectively these microbes can be utilized. And the third one is the crop nutrition. Yes, nutrient use efficiency. We have today excellent deposits of chemicals in the soil. and all are deposited, not available to the plant. Therefore, these microbes, they produce organic acid, dissolve these nutrients, chemicals, convert into nutrients and supplies to the plant. So likewise, nitrogen fixation, carbon sequestration, phosphate solubilization, there are plenty of essential, functions of the microbes helps crop productivity. So, thank you so much. This is flash to field to feed the future. Thank you so much. I think we have very little time left for panel discussion. So I think I have only 2 sets of questions. So this side, all the microbial team, and that side is organic team, I can say. And so each team will get only 1 question, and we will wrap up in maybe 6 to 7 minutes. So Professor Appara Garundu, Dr. A.K. Saxena, the question is, so what are the innovative approaches from the microbial side, and what kind of a message you are going to give to the industry, and what are the avenues? Please.
Now, from the microbe side, what we need to understand is more detailed microbe interactions and microbe-plant interactions, because if we have one important thing that we have missed out is when we look at the ancient genotypes of the crop, they are better suited for colonization by the microorganisms. Over a period of time during breeding or domestication, the plants have lost the capability to associate with the microbes as much as the older genotypes can do. This has been established. And now the breeding strategy, breeders, not only our group people sitting here, breeders also have to be components of, or to be conscious of looking at the microbiome when they are looking at the newer varieties that are to be coming up. And when we are talking about carbon fixation, in fact, that was one slide which I have not explained more. When we are talking about carbon fixation, there are ample scopes which have come through modern science these days. At least 28 different pathways are possible to fix the atmospheric carbon, other than just the 2, the C3, C4 pathways that we know today. And in microbes, we know about half a dozen pathways. But 28 different possibilities have already been identified. When we breed or when we have the next generation crops which can fix more of carbon, they can send more of carbon, they can be tailor-made to secrete more of carbon into the soil and facilitate more of microbial interactions. This is a combination effort that is to be done by the microbiologists and those who are working with microbes to ensure that we have a better synergy between these organisms and the plants for us to have more and more meaningful exploitation of the plant microbe interactions. This is something I wanted to add.
See, I would be a bit offline. When we are talking about the microbials, just like the soil health card, I think we should have the microbial health card also of the country. We should know what are the keystone taxa which are important for the sustenance of the ecosystem. We should identify those keystone taxa. And when we are talking about the microbiome, look for the possibility, can the microbiome can also be cultured? We can culture single organism, we can make consortia. But whether we have ever thought of multiplying the microbiome, I would just like to give an example. Valley of Flowers, I think many people of you must have visited. It is one of the self-sustaining ecosystem. Nobody is giving any fertilizer there, nobody is giving any irrigation there. But still, every year, you will find the valley full of flowers. The plants are blooming. So, such type of self-sustaining system, can we get the microbiome from there, and can we have the technology to multiply it? If we can have that type of a technology, We have to go away with the single formulations, the consortia. We can only go for the useful microbiome, which can be utilized for any cropping system. So this is a big question and a very ambitious, I think, for the industry as well as for the academia also. So this is what I wanted to mention.
Thank you, Dr. Tanmay.
Sorry, sir, could you?
So what message would like to give to the industry and what suggestions out of your experience to take it forward the marine science?
I think the opportunity in the seaweed space is just starting. There is a lot of work to be done on algae. I think for India to succeed, based on our experience, the biggest challenge is bringing in of new varieties, sustaining those varieties, adapting them to Indian waters, convincing the government what species are safe. There are misnomers about what is invasive, what is not invasive, what can adapt, what cannot adapt. And a lot of experts here have the experience, knowledge, background, tissue culturing, techniques, going deeper into the cells. How to do that in India and how to make us a seaweed producer successfully is a huge area of opportunity. There is finally a ministry behind it. There is funding behind it. So if the project has merit, it's the time to act. And I think it's a sunrise sector, so we can all grow it together.
Thank you, Tanmay. Roger?
So my point, all of this should be inclusive approach. We should not just talk about in isolation only for the microbes or seaweed. I think those days are gone when we were talking about excluding something. In my opinion, and that's the message I try to give everywhere, we should have an inclusive approach. Start from the farmer and the crop and look at what the farmer and the crop needs. Because in every country, within the country, there are so many regions where you can find the solution from different means. So we should try to look at not just for selfish reasons, but from collective, what can help the industry in general. That's number one. Number two, what I have also seen that we have to, just like I'm totally converted on microbes since yesterday, guys. So thanks to all of you, the work you have done in India is mind-boggling. Second approach, I always say, we have to make it more global. We have to take it from India to global and global to India. And that's the most important part for us, these two things. Last one is the soil to shelf, as you also mentioned, that we have to look at the solution for the BioAg from soil to shelf. Thank you.
Thank you, Roger. Thank you very much, speakers, and hope the session is interesting to all of you. So give a big hand to the speakers, please. Thank you so much.